All right, welcome to INBDE Microbiology Review number 6. All right, make sure you subscribe so you can receive notifications to the new videos. All right, so let's discuss bacterial metabolism. Okay, so different sub-classifications of metabolism. You have catabolism, which means to cut in pieces, to break down, and anabolism, okay, or anabolic pathways to build, such as making the carbohydrates, making carbohydrates into glycogen, okay, or making taking fatty acids and making them into lipids. Okay, that's anabolism. Catabolism is to break down. All right, and there are two forms of energy currency for bacteria. You have ATP, okay, and you have electrochemical gradient, which is also called proton motive force, where bacteria produces these protons, hydrogen ions, and then they use that for energy due to the gradient. Okay, and there is usually interconvention of energy forms, okay, by membrane ATPase. So, derivation of energy from carbohydrate metabolism occurs through glycolysis and fermentation. Okay, if you want to review the glycolysis, there's the lectures in biochemistry which are broken down. Okay, for glycolysis, you have to understand glycolysis and fermentation. Okay, if you're going to take this exam, so make sure you go and review the glycolysis lectures. I think there are 4 of them and discusses the basics of what you need to know about the glycolysis. Okay, all right. So glycolysis, glucose in glycolysis, meaning the breakdown of glucose. Glucose is converted into 2 molecules of pyruvate with a net formation of 2 molecules of ATP. All right. And then there's substrate level phosphorylation. Substrate level phosphorylation, ATP is produced by direct transfer of phosphate from 2 glycolytic intermediates to ADP. Okay, so you transfer the phosphate to ADP, you make ATP. That's substrate level phosphorylation. Okay, all right. So fermentation. So fermentation, you need to understand that there's in fermentation generates NAD, so NAD plus, which is reduced to NADH. Okay, so NAD is reduced to NADH during glycolysis. Okay, and fermentation products vary depending on the species of bacteria and can be used in the identification of bacterial species. For example, in the table above, Streptococcus mutans, okay, produce lactic acid. Lactobacillus, these are key bacteria in which produce caries, okay. Lactobacillus also produces lactic acid. Now, if you see formic acid is produced by enterobacteria, okay, and butyric acid is produced by Clostridium. So fermentation produces different products based on different species of bacteria. All right. How about aerobic respiration? Okay. So aerobic respiration is the use of oxygen, okay, to produce ATP. And in aerobic respiration, oxygen is the final electron acceptor. That is the purpose of oxygen. We need oxygen to accept the electrons from other molecules. Okay? So, most organisms perform glycolysis or fermentation. Okay? If you have oxygen, then you're going to undergo either the citric acid cycle, and you're going to undergo the oxidative phosphorylation. Now remember that oxidative phosphorylation occurs in the mitochondria, all right, via the proton gradient. Okay, now the energy yield in respiration, you have 34 ATP overall, and two ATPs, remember, are produced from glycolysis. Glycolysis produces 2 net ATP, and I say net, okay, net ATPs, and you have 2 ATP from the citric acid cycle, and this produces 38 ATP. Okay, so 38 ATP molecules are produced from one glucose molecule. So how many ATPs are produced from one glucose molecule in aerobic respiration? Overall, 38. Okay, that is in bacteria. So we're talking about the bacteria here. All right, 38 ATP per glucose molecule in aerobic. So this is obligate aerobes, the bacteria that need oxygen. Okay. All right. So oxygen utilization and tolerance. Remember, some bacteria uses oxygen, some they cannot tolerate oxygen whatsoever. Okay. So enzymes required to detoxify oxygen by byproducts are: one is superoxide dismutase. This is very critical. Superoxide dismutase catalyzes the breakdown of the superoxide, okay, the dismutase molecule, which is an oxygen molecule with an extra electron. All right. And also the other enzyme that participates in detoxifying oxygen, okay, species is peroxidase, which usually breaks down glucose, okay, reactive oxygen species into hydrogen peroxide. Okay. All right. So how do bacteria uptake nutrients? Okay. So usually involves active transport. Usually active transport means it needs energy. Okay. Active needs energy, and there's also protein driven uptake of nutrients, ATP driven transport, and group translocation. This is how bacteria takes in nutrients. And classification of organisms based on oxygen usage and sensitivity, and we are going to break down each type. We have strict anaerobes. These strict anaerobes cannot tolerate to live with oxygen. So oxygen is toxic to strict anaerobes. And then you have aerotolerant anaerobes, meaning they can tolerate some amount of oxygen, okay? And then you have strict aerobes. Strict aerobes need oxygen to survive, okay? And then you have facultative bacteria. Facultative bacteria can either live without oxygen or with oxygen, okay? And then you have microaerophilic bacteria. Okay, so let's discuss each one of them. All right, so strict anaerobes cannot grow and some cannot survive in oxygen. Okay, usually due to lack. So strict anaerobic bacteria, they lack these enzymes which detoxify the reactive oxygen species. So they lack superoxide dismutase or catalase. Okay, so this here, catalase is spelled, it's missing an A. It's catalase, like this. Okay, all right. So there's lack of respiratory chain enzymes. Okay, so they lack respiratory chain enzymes, and these bacteria depend on fermentation. Because remember, if you don't have oxygen, what else were you going to do? You're going to ferment. Okay, so they rely on fermentation. So strict anaerobes, fermentation. They cannot survive in oxygen. Okay, now aerotolerant anaerobes, they tolerate oxygen, but they do not use it. Meaning, oxygen doesn't kill them, but they don't use oxygen because these are anaerobes, okay? And they lack respiratory chain enzymes. They also depend on fermentation. So aerotolerant versus strict anaerobes. So strict anaerobes cannot tolerate oxygen, but they depend on fermentation. The aerotolerant anaerobes, they can tolerate oxygen, but they don't use it, and they also depend on fermentation. How about strict aerobes? So strict aerobes, they must have oxygen. Okay, it's like people. We must have oxygen to survive. Okay, so the strict aerobes are restricted to using aerobic respiration. This is people. We need aerobic respiration. We need that oxygen. Okay, so these bacteria, they also need oxygen to survive. They can only use oxygen as final electron acceptor. Okay, and strict aerobes lack enzyme for fermentation. So just like people, we do not ferment anything. Okay, we only produce lactic acid when we are running when we are running and we don't have enough oxygen as supplied to the muscles. Okay, however, we do not have enzyme for fermentation, or that's what strict aerobes do. So make sure you know the different types of bacteria and how they use oxygen, or how some of them cannot tolerate oxygen. Okay, how about facultative bacteria? They are capable of aerobic respiration in the presence of oxygen, and they use either anaerobic respiration or fermentation in the absence of oxygen. So these guys are the survivors. Okay, they're the ones which say, okay, you give me oxygen, I'll be fine. If you don't give me oxygen, I'm still fine. Okay, because they can use either oxygen or they can live without oxygen. Okay, so they don't care whether they have. So these are the dangerous guys. Okay, because they can live with oxygen, but they can also survive without oxygen. All right, and then there's a microaerophilic bacteria. These are bacteria which require a small amount of oxygen, but too much of oxygen, it becomes toxic to them. Okay, all right, so make sure you review this type of bacteria. Make sure you review the series number 1 through 6. Okay, and our next one is going to be the review series number 7. All right, if you have any questions, again, as I mentioned, is you comment or you email me. Okay, some of you have been emailing. Keep the questions coming. All right, good luck.